Hello, hello. Today we are joined by a very special guest, Dr. Keng Chou from the Department of Chemistry at UBC, to discuss his revolutionary high-resolution microscope. Welcome back to our podcast. It's Darren,
Chantelle, and Sarah.
So what got you interested in doing research?
When I was a kid, I just liked reading science book. So when I started to read, I read a few books, and I found I myself like science book more.
Mm-hmm.
And you keep reading, and I still remember the first book I read about atoms. Right, and I realized if you look at an atom, for the first time I understand the structure of atom. There is a very small nucleus at the center, and the electrons are outside. So the first impression I got was, wow, I didn't know. In a material, it's mostly empty space. All right, because you look at atom, electrons are very small, nucleus are very small. So if I have a very large, very good microscope, I zoom into atom, it's actually mostly empty. So myself originally do spectroscopy. Spectroscopy study interaction between material and light. And eventually microscopy is also interaction between light and material. It just helps us to see better.
I see. So you're always interested in microscopes, but we were wondering what makes your microscope so special compared to other ones?
Yes, so right now our microscope, we have 3 microscopes in the basement of Life Science Institute.
And they were developed by you?
Yes, we built all of them. So each one have different functions. One of them has very high resolution, 10 nanometer. The other one, the resolution is 100 nanometer, but we can do 100 frames per second. So we want high speed, high resolution imaging. And then, the size of virus is about only a few hundred nanometer. So regular optical microscope would just see one spot. Because regular microscope have a resolution of 200 nanometer. So it basically just one pixel.
Right. Is there any way you can build a more concise microscope?
What do you mean concise?
I meant
higher resolution?
Higher resolution.
So 10 nanometers is the current limit. If you want something even higher resolution, you have to go to electron microscope.
Have you thought of entering that field?
Electron microscope is a different field, because the problem with electron microscope is you have to fix the cell. You cannot do live cell electron microscope. Right. So in optical microscope, I can image a live cell. So this is like if I watch a football game. Assuming you don't know what a football game is, and you try to understand, and I only give you picture of a football game. For you, it would be very difficult to figure out what is the rules for the football game, right? But if I give you a video, then you will be much easier to figure out what is the rule, right? So I think the future, if you want to fully understand biology, live cell imaging is important.
I see. So how are you able to take photos at such a high speed if your 10 nanometer resolution microscope only takes images at 50 frames per second to create these live cell images?
In a conventional microscopy, I put in excitation light, all 404 will be bright. But if I can light up them one by one, then I can image them one by one and determine the center of the molecule one by one. So the way we do it, we take 40,000 frames. Each frame probably have 50 to 100 molecule. And we wrote a computer code, have computer analyze, locate we call blink. Locate the blink and fit the center of the blink, and then artificially put one dot at the peak of the blink. And if we collect a median blink, then in a image, I will get 1 million dot. Then that give us the 10 nanometer resolution.
It's all very interesting. After a short break, we will have Dr. Chou.
No. I already ate a lot.
Yeah.
All right then. Thank you so much for joining us here in our studio today. We appreciate your time. Thank you all for tuning into our podcast today, and we hope to catch you all next time. Bye!